Welcome, everyone. Thank you for joining us for today's webinar, Standardization of Cell Viability Assays in Primary Cells as a Prerequisite for Novel Bioprocessing Applications, presented by Dr. Simon Kaja, Assistant Professor of Ophthalmology and Molecular Pharmacology and Therapeutics, Loyola University of Chicago. My name is Xavier Gutierrez, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Kaja. I will now turn the presentation over to him.
Hello, everyone, and thank you for joining this presentation. And thank you, Xavier, for the kind introduction. Before I begin my presentation, I'd like to make you aware of two financial disclosures. I'm shareholder and board member of KNP Scientific LLC, and also shareholder and member of the executive leadership team at Experimentica Limited. However, I will not be discussing any commercial applications today, so there is no conflict in any components of the presentation. To start off with, let's consider the bioprocessing pipeline for medicinal formulations. Traditionally, the pipeline will begin with cell line development, be followed by upstream processing, downstream processing that often involves any kind of purification, etc., the formulation development, and ultimately the process analytics leading to a therapeutic. In today's presentation, we will focus on the first component highlighted here in the red box, the cell line development. When we think about medicinal bioprocessing, there are a lot of what I would term classic applications that we're well familiar with. Many of these involve suspension cell lines, such as Chinese hamster ovary cells or CHO cells, and they're used for the production of many protein-based drugs. Some prominent examples here are factor VII for blood clotting, insulin, and many others. More recently, we've also seen an advancement in the generation of antibody-based drugs, where suspension cell cultures are used to synthesize and purify immunoglobulins. However, new advances in the research and science have led to novel applications that require bioprocessing applications. One such example is a generation of exosome-based medicines, and I'll talk a little bit about exosomes in the next couple of slides. This includes also personalized medicine therapies that rely on stem cells, as well as other specialized cell types, such as astrocytes and neurons. So we see a need for primary cell culture and the following optimization for downstream processes. One example I'd like to give you today is the generation of bioengineered exosomes. Exosomes, or extracellular vesicles, EVs, are secreted from essentially all cells. They're very small membrane-bound particles in the nanometer size range, and they carry unique cargo. This cargo can consist of microRNA, RNA, DNA, protein, and many others. However, the critical component here is that exosome cargo depends on the cell type. So different cell types actually secrete exosomes or extracellular vesicles with very unique and distinct components. And it is this cargo or these components that are considered to exert the therapeutic effect. So if we're considering the use of exosomes and bioengineering them to contain a specified and predefined cargo, we need to optimize primary cell culture and the following downstream processes in order to make these extracellular vesicles or exosomes amenable to become therapeutics. My research group at Loyola University Chicago focuses on exosomes for the treatment of glaucoma. Glaucoma is a very prevalent neurodegenerative disease that affects almost 3 million people in the United States. Being an age-related disorder, together with an aging population, we anticipate that this disease will increase to a prevalence of over 4 million people by 2030. Of particular relevance, certain populations are of much higher risk to develop glaucoma. This includes African Americans of age 40 or older, and especially Mexican Americans, and essentially everybody over the age of 60. In glaucoma, the cell type that's affected are optic nerve head astrocytes. As you can see on the picture on the right, you see a fundus image of a healthy optic nerve, similar to what your ophthalmologist would see when he looks into a healthy eye. On the right part of the picture, you see an optic nerve in an eye that suffers from glaucoma. As you can see in the central portion, the white area is much disturbed, and this is the region where these optic nerve head astrocytes are located. Our approach is to trying to isolate exosomes and other and identify therapeutics that can target these specific cells. For this, we actually need to optimize the primary culture of these cells in order to establish cost-effective, high sensitivity, and high efficacy bioprocessing applications. This slide just gives you a quick overview over a typical culture procedure. In contrast to cell lines, where a cell stock is frozen down, primary cells have to be isolated from direct tissue material that can be obtained from an individual patient, or in the case of astrocytes, we can use donor or cadaver material to generate these in order to isolate therapeutics in the future. In this case, this is a rather laborious process of identifying and isolating the small region of the optic nerve head on the eyeball, which is depicted in the schematic diagram at the bottom, and then isolating these cells in primary cell culture, where they will grow out of the optic nerve and form a monolayer culture of high purity. One important component of optimization of these primary cell cultures are cell viability and proliferation assays. They really serve the purpose to determine the effect of treatments and also culture conditions on cell viability. This becomes important when we use drugs or therapies such as cytokines or oxidative stress to modify the exosome cargo that is secreted from these cells. Also, cell viability can be affected by the cell media and supplements used to grow and maintain these cells. We can also use cell viability assays to quantify the rate of proliferation, which is of critical importance for the scalability and efficacy of bioprocessing. Two of the most popular and widely used cell viability and proliferation assays are the MTT assay and the lactate dehydrogenase assay. The MTT assay, and MTT stands for 3,4,5 dimethylthiazole 2-ol-2,5-diphenyltetrazolium bromide, or short MTT, is based on the conversion of MTT to the colored dye formazan. MTT, when placed on live cells, can be taken up into the mitochondria of the cells, and inside the cells, an enzyme, mitochondrial reductase, will convert MTT to formazan. The purple product of formazan can then be detected spectrophotometrically. On the left, you see an example of primary cells that have been exposed to increasing levels of oxidative stress using a hydrogen peroxide analog, TBHP. As you can see, an increasing concentrations of TBHP leads to a decrease in the absorption. This reflects a loss of mitochondrial reductase activity in mitochondria and is a correlate of cell survival. Very similarly, the lactate dehydrogenase release assay is relying on a tetrazolium compound. In the living cell, lactate dehydrogenase catalyzes the reaction from lactate to pyruvate, and using NAD plus and NADH converts pyruvate to iodo-nitrotetrazolium, which is then converted to formazine using DAF arrays. In a living cell in the body or in organs, lactate dehydrogenase is secreted into the bloodstream. In cell culture, we can measure lactate dehydrogenase in the media supernatant. Therefore, the LDH release assay has the advantage that it can be used for both adherent and suspension cell cultures. Furthermore, it can be very readily adopted to primary cells. But why optimize LDH release assays? It's a well-established assays, and there are numerous commercial solutions. There are standard protocols for many routine applications. However, one of the major drawbacks of commercial LDH release assays is that they're very costly, and they also have limited options for optimization. What I will present you today is the development of a custom LDH assay. It has known components in a very small number, and thereby provides greater options for optimization and the flexibility to adjust the assay to your specific experimental needs. In addition, it is 20 to 30 times cheaper than commercial options. And in addition, the assay is readily scalable from our typical 96 well assays all the way to true high throughput applications. Our group has published several manuscripts describing the LDH release assay that are listed here. The first one initially validating the assay on cell lines, then moving it to primary astrocyte culture, and in the last article providing detailed step-by-step protocols, including troubleshooting tips for primary astrocyte cultures and primary neuronal cultures. Later in this presentation, I will discuss both the individual steps as well as some of the troubleshooting that can occur with these assays as it is being adapted for primary cells. We have designed our custom LDH release assay to contain a minimum of solutions and reagents. Essentially, there are 4 solutions that are needed. Buffer A and buffer B are combined in equal volumes and contain all the components to catalyze the reaction. The supplement is added in order to provide the color reaction, and having the supplement as a separate reagent allows the titration for very small or larger amounts of lactate dehydrogenase that is being released into the particular media for a given application. The stop solution is merely 1 molar acetic acid that serves to stop the reaction and provide ample time for subsequent analysis. For a 96 well plate-based assay, the diagrams depict the typical workflow of adding a drug or any kind of media, letting the cells incubate for varied amounts of time, anywhere from a few hours to overnight, then collecting the supernatant in a new plate, adding the lactate dehydrogenase assay components that have been pre-mixed and consist of buffer A, buffer B, and the supplement, and then following adjustment and optimization of the incubation time, which is typically anywhere between 30 minutes and 2 hours, stopping the reaction with acetic acid and measuring the absorbance at 490 nanometers wavelength. Here I'm showing you our initial validation of our custom assay compared to the Cytotox 96 assay, which is a commercially available solution. The cells we used in this one were initially a cell line, C6 astroglioma cells that we treated with oxidative stress. In the gray lines, you can see the untreated cells, and in dark gray cells treated with the vehicle, which in this case was 0.1% ethanol. In orange, you'll see cells that were also treated with an antioxidant, Trolox, which is a prototypic vitamin E analog and very potent superoxide dismutase scavenger. On the x-axis, you see increasing concentrations of tert-butyl hydrogen peroxide and more water-stable analog of hydrogen peroxide. As you can see, as cells suffer from oxidative stress and reduce their viability, you see a significant increase in LDH release, which can be potently prevented by Trolox. As you can also appreciate, there is no difference in sensitivity or efficacy of our assays compared to the commercial situation. In this graph, I've overlaid the untreated cell graphs for our custom assay as well as the commercial assay. And to put the pricing and the scalability into perspective, while our custom assay on average costs about $1.20, commercial solutions typically average of $30 to $50 per plate, providing a significant cost saving, which is in addition to the advantages of better flexibility in terms of optimization and scalability. In our article from 2017, we've also provided protocols how this assay can be adapted to cultured rat cortical neurons, as well as to primary cultured optic nerve head astrocytes. Again, here you see untreated cells, which are depicted in the red line labeled control, or treated with Trolox as an antioxidant. What you can appreciate is a right shift in the curve, making the cells less susceptible to the deleterious effects of oxidative stress. For bioprocessing, one important consideration is the optimization of media formulations. Different medias are required, and different media can contain different formulations, different amino acids, different amounts of sugar. And primary cells are much less resistant to exogenous stresses as many cell lines. So here it is important to actually optimize the media formulation, and the lactate dehydrogenase release assay provides one very simple option to do that. What I'm showing you here is an analysis of lactate dehydrogenase release compared to a control media for 10 different media compositions. And as you can see, some of the medias, for example, 3 or 6 or 10, show significant levels of lactate dehydrogenase release. This is an indicator that cells are undergoing apoptosis and are dying, or at least are stressed. This can have deleterious effects on any subsequent bioprocessing application. So optimizing your cell culture using a media formulation that is suitable to the cells and provides maximum survival will provide much better efficacy in the downstream bioprocessing applications. Furthermore, the lactate dehydrogenase release assay can be used for toxicity screening. One example is the traditional toxicity testing for the development of nutraceuticals. However, as we're talking about exosomes and utilizing exosome populations to serve as therapeutics, supplementations of cells with nutraceutical compounds or antioxidants or any other drugs can modify, for example, the antioxidant potential of these cells and thereby create a unique cargo in our exosomes. Here I'm showing you an example of using human corneal epithelial cells that were treated with xanthomumol. Xanthomumol is a plant-derived antioxidant that occurs in the hops plant. In the top panel, labeled A, you see an MTT assay. With increasing concentrations of xanthomumol, we're seeing a loss in cell viability at high doses of about 50 and 100 micromolar. This is reflected and very well tested with the lactate dehydrogenase assay. I'm comparing here both the lactate dehydrogenase assay and the MTT assay. The advantage of the MTT assay is that it is suitable for both adherent as well as suspension cell lines, whereas the MTT assay is traditionally used in adherent cells, which makes it less flexible for bioprocessing application. However, what I hope you can appreciate is the fact that both assays provide very similar results, making the lactate dehydrogenase assay, given its flexibility and cost and options for optimization, the preferred option. Also, the lactate dehydrogenase release assay, the way we've developed it, provides a lot of options for troubleshooting and optimizing it to your specific needs and the specific cell line or primary cell that you're working with. I want to go through some of the problems, their causes, and their solutions, as these are very universal and can be applied to very many different cell types. Obviously, the biggest problem is if there is low or no signal. That is often related to very low density that can occur in early bioprocessing before optimization has taken place. Obviously, the solution here is to either increase the seeding density or to have a higher concentration of cells per volume. Also, test compounds or media may inhibit lactate dehydrogenase activity. Given that our assay contains so few reagents, it makes it very easy to actually test in a standardized setting that might be cell-free to actually see whether any component interferes with the enzyme activity itself. Media optimization is also important, as many media that we're using in bioprocessing applications contain sodium pyruvate. Sodium pyruvate is a very well-known inhibitor of lactate dehydrogenase. So therefore, avoiding media with sodium pyruvate is often necessary to prevent any confounding effects when conducting the LDH release assay. Also, the incubation time for the assay might be too short, and it's easy to conduct a kinetic run and monitor LDH release over time. An example of such experimental design is provided in our publication from 2015 that's referenced here. You will also find these tables in our publication from 2017 that I've referenced on the earlier slide that I'll reference again on my final slide. Another problem that is often encountered is a too high background signal or a signal that is being saturated. Obviously here, the cell concentration might be too high, and as in the previous step with too low signal, optimizing seeding densities can resolve this problem. However, sometimes the intrinsic lactate dehydrogenase activity in the serum or in the media is high. What's possible here is to reduce the serum concentration, or often readily achieved by reducing the concentration of the supplement. Typically, we use the supplement at a 1 in 10,000 dilution, but it can be easily diluted to 1 in 20,000 or 1 in 50,000. Also, heat inactivation may affect the serum LDH activity. Typically, we would recommend testing the endogenous activity of the serum by performing a cell-free or serum-free and media controls. Complete medium LDH activity is typically similar to the observed LDH activity of untreated cells, so that makes the experimental design very easy. Also, the target signal might be too high or saturated. This is often a problem with commercial assay, where there's limited options for optimization. Again, here, our assay allows to adjust the concentration of the PMS supplement. Again, as previously, it can be readily diluted to 1 in 20,000 or even 1 in 50,000, which is a much simpler solution than having to adjust the workflow and diluting the precious sample material that is being used for the assay by doing another step that would require standard operating procedure. Rarely, the dye might turn yellow after addition of the stop solution. This typically indicates that the concentration of acetic acid is too high. The reduced dye, which is normally purple for formazan, can spontaneously oxidize back to its initial reactant, which is light yellow, when the pH reaches levels of lower than 4. You can either reduce the amount of stop solution and, of course, easily test the pH after adding stop solution using a pH strip or pH meter, and ensuring that the pH stays above 4.0. So in summary, novel bioprocessing applications may require the use of primary adherent and suspension cell cultures, such as astrocytes, fibroblasts, and neuronal cells. Adaptation of bioprocessing to the use of primary cells requires optimization. Optimization of culture conditions for different cell types is critical, and primary cells have very different requirements and cell lines, making this a much more challenging task. This optimization requires assay development for the use of cell viability and proliferation assay. For this, the lactate dehydrogenase release assay is a well-established assay that can quantify very subtle changes in cell survival, which is critical in identifying potential confounding problems when utilizing these for future bioprocessing and downstream applications. Our LDH assay can be developed as a very cost-effective and customizable assay. It consists of known components. It's got a strong flexibility for optimization, as I've shown you in this presentation. It is about 20 to 30 times cheaper than commercial solutions, and it is very readily scalable. Again, down here, I've listed the 3 publications that I've referred to in today's presentation, together with the PubMed ID numbers, so you can easily retrieve them on PubMed. Lastly, I'd like to acknowledge the funding sources that have supported the research in my lab over the last few years, include the Glaucoma Foundation, the John and Therese Mulcahy Endowed Professorship in Ophthalmology at Loyola University Chicago, several other of our foundation partners, the Department of Veterans Affairs, as well as Experimentica Limited. Experimentica is a contract research organization, so we work very closely in assay development. And also thanks to my collaborators here in Chicago, in Kansas City, by name of Peter Kuhlen, and Experimentica in Finland. And of course, none of this work could have been done without the excellent lab members and staff members that I have in my group. And I'd like to acknowledge particularly Anita Ghosh and Harsh Haryani, who have contributed a lot to the work I've shown you today. Again, thank you very much for your attention and for your interest in today's presentation. And I look forward to receiving your questions and answering them by email. Thank you very much.
Thank you, Dr. Kaja, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. Now, before we go, I want to let everyone know that this webcast will be available for on-demand viewing through April of 2019. As a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. We hope to see you again soon. Thank you. Goodbye.